Hello student, this is Pratibha and today I am going to give a lecture on biological classification from class 11th, chapter number 2. This is a very useful video for all the students who are preparing for the competitive exams. So if you like this video, please share, like and subscribe this video for all my educational videos. So you know the dawn of civilization, there have been many attempts to classify living organisms. It was done instinctively, not using criteria that was scientific, born out of a need to use organism for our own use, for food, shelter, and clothing. Aristotle was the earliest to attempt a more scientific basis for classification. He used simple morphological characters to classify plants and animals. So there is the classification of Aristotle. He classified plants to trees, shrubs, and herbs and animals into two groups: those with red blood and without red blood. Red blood includes mammal, lizards, and birds, and no red blood include hard-bodied and soft-bodied. Hard body have insects, and soft body included shell and no shell. Shell is a shellfish, and no shell is a jellyfish. After it, a new kingdom classification was arised, that was two kingdom classification, and it was proposed by Linnaeus in 1758. The system classifies organism into two main kingdom, that was Plantae and Animalia. But there are some drawbacks of this two kingdom classification, that is prokaryotic like bacteria and cyanobacteria, and eukaryotes like fungi, mosses, ferns, gymnosperms and angiosperms were included under plants based on the presence of cell wall, but they are widely differed in other features. It included unicellular and multicellular organism in same group. Example, Chlamydomonas and Spirogyra were placed under algae. And the third is it did not differentiate between heterotrophic fungi and autotrophic green plants. Fungi have chitinous cell wall while the green plants have cellulosic cell wall, and it is a very different from each other. So after this drawbacks of two kingdom classification, a new kingdom classification was arrived, that is the five kingdom classification, and it is proposed by R.H. Whittaker in 1969. The main five kingdoms was Monera, Protista, Fungi, Plantae, and Animalia in the five kingdom classification. And this classification is based on cell structure, thallus organization, mode of nutrition, reproduction, and phylogenetic relationship. So there is a chart for this five kingdom classification by using some characters. Like first is the cell type. Monera is a prokaryotic type, but Protista, Fungi, Plantae and Animalia are eukaryotic type. Cell wall. In the Monera, non-cellulosic cell wall is found, which is formed by the polysaccharides and amino acid. Protista have sometimes cell wall is found. Fungi, there is chitin is present in the cell wall. In the Plantae, cellulose is found, but in Animalia, cell wall is absent. Next is the nuclear membrane. Nuclear membrane is absent in Monera, but it present in Protista, Fungi, Plantae and Animalia. Next is the body organization. In the Monera and Protista, you can see cellular organization is found, but in fungi, multicellular and loose tissues are found. In the Plantae, tissue and organs are found, and Animalia, tissue, organ and organ system is completely developed. Next is the mode of nutrition. In the Monera, autotrophic like chemosynthetic and photosynthetic, and heterotrophic like spirophytic and parasitic. Protista are autotrophic type, it means these are photosynthetic and heterotrophic type. Fungi is a heterotrophic, it includes saprophytic and parasitic. Plantae is a autotrophic, it includes photosynthetic activity, and the Animalia are heterotrophic, holozoic and saprophytic type. So we come to the main kingdom, first main kingdom, that is the Monera, and it include all the bacteria. In the first kingdom Monera, bacteria are included. As you know that bacteria are the most abundant microorganisms which are found in everywhere. Hundreds of bacteria are present in a handful of soil. They also live in extreme habitats such as a hot spring, deserts, snow and deep oceans. They are many times they are parasites. So we can classify bacteria on the basis of their shape into the four groups, that is spherical coccus, rod shaped bacillus, comma shaped vibrio and spiral spirillum. So there are some characteristics of the bacteria. Some of bacteria synthesize their own food from inorganic substances. These are autotrophic, like photosynthetic autotrophic and chemosynthetic autotrophic. Some bacteria do not synthesize their own food but depends on other organism or on dead organic matter for food. These are heterotrophs. On the basis of some characteristics, we can classify bacteria into two groups, that is Archaebacteria and Eubacteria. Archaebacteria have some specified characteristics. These bacteria live in the most harsh habitats such as extreme salty areas like halophiles, hot springs like Thermoacidophilus, and marshy areas like Methanogens. Archaebacteria differ from other bacteria in having a different cell wall structure, and this feature is responsible for their survival in extreme conditions. Methanogens are present in the gut of several ruminant animals such as cow and buffaloes, and they are responsible for the production of methane biogas from the dungs of these animals. Second is the eubacteria. There is some characteristics of the eubacteria also. There are thousands of different eubacteria or trubacteria are present. They are characterized by the presence of a rigid cell wall and, if motile, a flagellum. Some of these organisms can fix atmospheric nitrogen in specialized cells called heterocysts like Nostoc and Anabina. They play a great role in recycling nutrients like nitrogen, phosphorus, iron and sulfur by using nitrogen cycle, phosphorus cycle, iron cycle and sulfur cycle. So it is a very important point. Next is the heterotrophic bacteria are the most abundant in nature. The majority are important decomposers. Many of them have a significant impact on human affairs. Sometimes they may be beneficial and may be harmful. They are helpful in making curd from milk, production of antibiotics, fixing nitrogen in legume roots, etc. Some are pathogens causing damage to human beings, crops, farm animals and pets. Some diseases causes cholera, typhoid, tetanus, citrus canker in lemon are well-known diseases caused by different bacteria. There is some characteristics of the bacteria that they grow mainly by fission process. Sometimes under unfavorable condition they produce spores. They also reproduce by a sort of sexual reproduction by adopting a primitive type of DNA transfer from one bacterium to the other. So there is a picture in which eubacteria is classified into two groups, autotrophic and heterotrophic. Autotrophic is also divided into two parts, photosynthetic, example blue-green algae, Nostoc and Anabina, chemosynthetic, Vestimentifera and the Cryptogena. In this diagram we can see Nostoc, a blue-green algae which have heterocyst for the photosynthetic activity. So there is another group of bacteria is present, that is the Mycoplasma. The Mycoplasma are organisms that completely lack of cell wall. It is a very important question. They are the simplest free-living prokaryotes. They are also known as PPLO, pleuropneumonia-like organism. Very important for the competitive exams. They are the smallest living cells known and can survive without oxygen. Many Mycoplasma are pathogen in animals and plants. And there is a diagram for a dividing bacteria in which we can see by the fission process a cell is dividing into the two cell, and the cellular material, that is DNA, is also dividing by this process. So thank you so much for your kind attention, and I will come with my second part of this chapter very soon. So if you like this, please subscribe my channel for many videos which are very useful for you, and if you want to ask any questions, please comment in comment box. Thank you so much.